आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome to the podcast from Indo French Center for the Promotion of Advanced Research, or CEFIPRA. We are here to showcase the exciting collaborations between Indian and French scientists, spanning a wide range of scientific fields from fundamental research to applied sciences. Today, we are spotlighting a project that exemplifies CEFIPRA's mission of strengthening scientific ties between India and France. It brings together Gayathri Pananghat from the Indian Institute of Science Education Research, IISER Pune, and Professor Tam Mignot from the CNRS Laboratoire de Chimie Bacterienne to explore a fascinating biological process, how the bacteria Mycococcus xanthus reverses its cell polarity. Mycococcus xanthus is a rod-shaped bacteria commonly found in soil. What's remarkable about it is its ability to frequently switch its forward end, a process called cell polarity reversal. This allows bacteria to move in a coordinated fashion and form a complex structures called fruiting bodies. Imagine a bacterial ballet where cells change its direction in a synchronized dance. To understand this intricate process, researchers in India are conducting detailed laboratory experiments studying the molecules involved. Meanwhile, in France, scientists are using advanced imaging techniques to observe these bacteria in action, directly visualizing how they move and organize. Now, let's listen to them.
I am Gayathri Pananghat. I am an associate professor at the Indian Institute of Science Education and Research, Pune. I am a structural biologist by training, and our lab addresses problems related to understanding bacterial shape, motility, and cell division. I am a structural biologist by training, which means that I look at structures of biomolecules. So these are typically protein complexes which I study. So we decipher the positions of atoms, of each individual atoms of the protein molecules, and through the structures we try to get an idea of the function. So the problem which we are addressing in our lab is about protein complexes which are involved in determining the shape of bacteria, also looking at the proteins which help in cell division of bacteria and proteins that help bacteria to move. So that's the problem which we are addressing. And how do these molecules drive the biological process is what we are looking at. The model systems which we are using to address this question are bacteria so there are two systems which we have chosen. One is a soil bacterium, which is Mycococcus xanthus, which glides on the soil surface. What we are looking at is to understand the process of how the bacterium glides on the surface. Essentially, how does the bacterium move and reach various places so that it can survive as a community. And the significance of using bacterial systems or studying bacterial motility, shape and division is that you can, first of all, in bacteria, these model systems which have been chosen are what could be considered as the minimal machinery or the smallest unit at which we can get a mechanistic information. Another significance in studying these is we can also look at novel targets for which we can develop antibiotics so that these bacterial processes are stopped for pathogenic bacteria. So this is the long-term goal of the research as such. So it understands fundamental processes in biology, essentially how a bacteria moves and how processes like these help it survive in the environment where it lives. So in this particular project, which was funded by CIFIPRA from 2018 to 2021, we looked at the Myxococcus xanthus motility. Myxococcus xanthus is a soil bacterium and it moves on the surface, as I just mentioned. And one of the interesting features of Myxococcus xanthus is it keeps shifting its polarity, or whatever is in the front and the back of the bacterium keeps oscillating. And this is driven by molecules. So you can imagine this as a train engine which moves on a train which moves on a track. If it has to reverse direction, the engine has to move from the front to the back. So that exactly is what happens in Myxococcus xanthus also. If the Myxococcus xanthus has to reverse direction, it changes from the leading pole to the lagging pole. This means that many of the components which form the engine, which help it to move forward, has to move to the other end. So that exactly is the process which we are studying which we studied using the project which was funded by CIFIPRA. This was in collaboration with a PI in France, in CNRS Marseille, Dr. Tam Mignon. So how we initiated this project is when I was starting the lab, I had decided to work on two of the systems, that's Myxococcus xanthus and Spiroplasma, for establishing my lab. At that time, the research publications which existed in the field of Myxococcus xanthus polarity was predominantly from Tam Mignon's group. And at a conference in 2012 on prokaryotic cell biology, even before I joined IISER Pune, I got an opportunity to interact with him and proposed his interest that I would like to work on this. Following that, I came back to India in 2013 and started my group. And in 2016 December, there was a conference in Trivandrum on bacterial morphogenesis and virulence, in which Dr. Tam Mignon was also attending. So we got a chance to interact at that time and also shared our results. And this was the point at which a CIFIPRA call was opened with January 15 as the deadline. And that's where we started putting up a proposal together in which I will be characterizing the structural work on the molecules involved in polarity oscillations, while the lab in France could contribute to the bacterial genetics. So one of the biggest advantages we had in this project was the complementary expertise. So their expertise was in doing in vivo work in Myxococcus xanthus, or actually showing how these molecules function within the cell, whereas our expertise was in the in vitro level, where we were looking at a molecular level to understand the system. So this collaboration is still ongoing because as the research work is progressing in both labs, there are many insights which are coming from both the groups, and we are in touch still for various other work which is ongoing in the lab. We also have a publication which is planned for regarding the complexes which help in deciding how frequently these bacteria change the rate of oscillation. So in future, as I mentioned, this is an ongoing collaboration, and we do plan to go ahead with more focus on the structural work for the multi-protein complexes which are involved in mixococcus oscillation. So the experience which I've had with the CEFIPRA funded project for this collaborative work has been excellent. So as part of the project, there were opportunities to travel to France. So I could avail the opportunity to travel to France to the collaborators group and initiate discussions on new work. In addition, I was also part of one of the meetings during the visit to Marseille. This was also clubbed with a meeting on women in science organized by CEFIPRA in Paris. In addition, this funding gave an opportunity to one of my students, my first PhD student who was working on the project, to spend about a month in the collaborators lab and carry out bacterial genetics work, which helped us to develop the project further. The support from CEFIPRA has been excellent, with very timely actions with respect to release of funds, and it has been a wonderful experience. In this context, I would also like to thankfully remember Mrs. Sati Devi, who was at CEFIPRA. She was the one who was contacting us throughout the project, and it's actually a sad event that she lost her life to COVID during the project. So she is someone whom I remember gratefully, and was very helpful to both to drive this collaborative project throughout, with suggestions on how to smoothly carry forward the administrative things related to the project.
The findings from this project will be shared through publication in top international journals, and where appropriate outcome through patents, potentially leading to new industrial collaborations. This is Harsha from CEFIPRA, signing off till we meet again with another project with another scientist.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान